All right, and I'd like to talk today about pattern completion, our ability to make inferences from partial information. Neil Burgess also discussed this in the context of forming episodic memories, and I think this ability to make inferences and complete patterns is a fundamental aspect of cognition and intelligence. And everything I'm going to talk about today concerns the work of two amazing grad students in the lab, Han-Lin Tang and Bill Lauder. We not only need to form patterns in order to build episodic memories, but actually I would argue that our ability to make inferences from partial information is a fundamental hallmark of intelligence that we use in a variety of different scenarios, from understanding sequences to actually being able to put together and connect the dots in order to understand and interpret music and many other sensory modalities. Even in social interactions, we can read social interactions from just a couple of seconds of basic information. And of course, my talk will concern our ability to recognize heavily occluded objects and studying pattern completion in the context of visual shape recognition. So we do this all the time. So here are a couple of examples of heavily occluded objects, and you can still kind of recognize what they are, despite the fact that most of the pixels or a lot of the pixels are actually missing. Of course, this becomes easier in the context of a whole image when you have context, and that helps a lot in terms of understanding what the objects are. But you can do so even in sort of almost very simple images where you take out all the context and you can occlude objects in a variety of different ways, several of which are illustrated here, including the one here illustrated in part F on the lower right, where you don't even need occluders to be able to make inferences about objects, and you can make these type of pattern completion tasks even with very, very small amounts of information without any context, without any occluders, without any stereo, without any other of these fancy cues. And I'd like to try to ask, what are the fundamental biological mechanisms that implement this type of pattern completion phenomenon? So I'd like to divide the talk into three parts. So this follows the star defined by Marr and Posho in order to understand any problem in vision, but I would argue any problem in cognition in general. We need to work at three different levels: at the behavioral level, at the neurophysiological level, and algorithmic level, which were roughly defined as computational, implementation, and representational levels. And I'd like to ask how pattern completion works at these three different levels. So let me start with behavior and just show you some data just to convey the notion that humans are extremely good at pattern completion and being able to make inferences about objects from very, very small amounts of minimal information. So we used a technique called bubbles, where we essentially are deleting parts of objects or looking at the world like this. Basically, you have a small bubble through which you look at the world. The more bubbles, the more of the object you see. Here's a toy school bus. When you have 20 bubbles, it's pretty easy to see. When you have 4 bubbles, it's pretty hard to recognize. So we can titrate the difficulty in the task. And we're going to use different levels of occlusion or visibility and evaluate how well people can recognize these objects. So here's the basic task. There's a fixation. There's a variable amount of time during which an object is presented. And then subjects have to make a 5 alternative forced choice categorization task to recognize these objects. And here's the behavioral data. Each of these colors corresponds to one particular SOA, one particular duration of the stimulus on the screen. And on the x-axis, you have the percentage of the object that's visible. So as you move to the right, you have more and more visibility. The task becomes easier. As you move to the left, there's a smaller amount of the object that's visible. Chance here corresponds to 20%. So you can see that performance is quite robust. Here, even if you essentially show up to 10% of the pixels in the object, people are still performing quite well. So it's only 1 in 10 pixels of the object essentially are necessary in order to be able to form the whole pattern, or at least to recognize in this 5-way categorization task what the object is. We can also do this not only with this bubble type of experiment, but also showing real occluders. The task becomes easier when you have real occluders, as illustrated here on the right, but the qualitative conclusions are essentially the same. We can still recognize objects in a very robust way, whether you have occluders or you don't. So let me now move into the brain and ask what are the neural circuits that are responsible for recognizing this type of heavily occluded objects. And to do so, we took advantage of a relatively rare opportunity that we have to investigate physiological function inside the human brain by virtue of collaborating with neurosurgeons who implant electrodes inside the brain of patients that have epilepsy. The whole purpose of this procedure is to treat the patients for epilepsy and to remove the part of the brain that's responsible for the seizures. This is a pretty well-established by now surgical procedure. It's been going on for decades. Nancy mentioned the work of William Penfield many decades ago, also with epileptic patients. And patients typically stay in the hospital for about 1 week. And during this 1 week, we have lots of electrodes in different parts of cortex. The location, number, type, and size of electrodes is not under our control. It's strictly dictated by clinical needs. But we have the unique ability to investigate human brain function at relatively high spatial resolution, very high temporal resolution, and importantly also with very high signal-to-noise ratio. So together with many other people, we've shown that there are visually selective signals in different parts of the inferior temporal cortex. These signals are similar to what Nancy has described using functional neuroimaging, as well as what Bob and many others have described using recordings in macaque monkeys. And I'm showing you one example of one of our recordings here, showing the selective response to pictures containing faces. On the y-axis, what you see is the intracranial field potential. This is raw data from one of our electrodes in the inferior temporal gyrus. On the x-axis, you have time. The trough of this signal, in this case, occurs at 150 milliseconds. Each of those gray traces corresponds to a single trial. The gray corresponds to the average of 39 trials where the subject was looking at this particular picture. So we see a very strong, visually selective signal. This signal is invariant to transformations of those pictures. I'm not showing that here. We've shown that in many cases, in many studies before. So what happens when you show these heavily occluded objects? When you only have about 10 to 20% visibility of the object, what happens with the signals from this electrode? So now I'm going to show you single trials from the responses of this same electrode. So here's one response to that particular picture, and you can see just in the raw data that even though we are only showing a very small amount of information, the response from this electrode is very similar to the one that we have for whole objects. Here are a few more single trials, and you can see that despite the fact that there is some heterogeneity from one trial to another, we can still see selectivity for this type of signal. And I'm pointing out here the latency of the response. What's the trough of the voltage as a function of stimulus onset? And you can see that in all of these single trial examples that I'm showing you, there's a consistent delay with respect to the latency that we observed for whole objects. In a couple of cases, we used a fixed position of the bubbles, so we repeated the same stimulus with the same position of the bubbles. Here are 4 trials with a fixed position of the bubbles, showing that there's a consistent response to those 4 presentations. Again, that's visually selective and delayed with respect to the presentation of the whole. Just to convince you that I'm not picking a few individual trials that looked nice, I'm going to show you all the trials and the responses from this electrode. So here are the responses to the whole images. Again, this is completely raw data. There's no maneuvering with the data here. Each of these lines corresponds to a single trial. The color corresponds to the voltage, and the x-axis here corresponds to time. There were 5 different phases presented. Those are divided by these horizontal lines and multiple repetitions of each of those phases. So you can see even with your naked eye that there's a pretty strong signal. There's something that happens in the voltage of this electrode in response to these whole objects. There's nothing tremendously new here. People have seen this over and over multiple times before. So now when we present those partial objects, the ones that where we only show about 10 to 20% of the object, again we see a strong visually selective signal that's connoted by these blue and red in these traces. You can see that there's a lot of heterogeneity in the signal. The pictures are different from one trial to the next because of the position of the bubbles. And despite this heterogeneity, there's a pretty strong visually selective signal in all of these cases. When we present the partial fixed condition, meaning that we have partial objects but we use exactly the same position of the bubbles, the signal becomes much more consistent, not as consistent as with the whole objects, but much more consistent. And we can see that in all the cases where we have partial object information, there's a delay of about 50 milliseconds or so in processing in the onset of this visually selective signal. We're particularly interested in these type of delays because they point to a significant constraint in our computational understanding of what's going on in terms of pattern completion. Bob already mentioned this in his multiple studies about attention and how different areas interact. In this case, we're interpreting these delays as implying that maybe there is a need for additional computations in order to be able to perform this type of pattern completion. So we conjecture that these these additional computations may involve the need for either top-down signals and or recurrent connections to be able to do pattern completion. So one of the sort of poor man's tools to investigate these recurrent and or top-down connections at the behavioral level has been the use of a protocol called backward masking. Essentially, you show a picture and you very briefly, very soon afterwards, you show a mask after that. And depending on the intensity of that mask and depending on the temporal contiguity between the stimulus and the mask, you can disrupt processing. You can either completely interrupt processing, or in some cases, you can essentially interrupt the feed-forward path according to many theories, as outlined here. So to the extent that backward masking interrupts additional computations, potentially interrupting recurrent and or top-down signals, we conjecture that backward masking would interfere with the ability to perform pattern completion. So we went back to behavior and we did a behavioral experiment with a backward mask. So here's the data that I've shown you before, showing the performance in object recognition as a function of what fraction of the object is visible. And here's the same case where now we introduce a mask at different SOAs, different stimulus onset asynchronous with respect to stimulus onset. So we see that if the SOA is pretty long, about 150 milliseconds or so, there's no impairment in performance. We recapitulate the results from the previous experiment. However, when the mask occurs within about 25 milliseconds or to 50 milliseconds of stimulus onset, we see that performance gets significantly degraded. So together with the physiological delays that I showed you in the previous slide, we take this as indication that there is a need for additional computations, perhaps recurrent and top-down computations, needed in order to be able to implement pattern completion. So here's the summary of behavioral performance as a function of the SOA. In the case of when there's no masking, this is the top line. And in the case where there is backward masking, and you can see how backward masking significantly disrupts behavioral performance. All of this is done with this bubbles experiment. Basically, the same results hold if you use real occluders, and that's shown in here. So now let me try to put together these two worlds. I told you about behavioral and the behavioral effects of masking. I told you that at the behavioral level, masking interferes with performance. I told you at the physiological level, there's a delay. And now I'm going to correlate these two worlds. Mind you, I'm going to do this in different subjects. We didn't do the backward masking experiment with the epilepsy subjects, so I'm going to correlate data from different subjects in this case. So I show you the behavioral, the physiological delay. So from that we can measure the amount of time that it takes to elicit a visually selective signal from partial images, and that's shown in here. I told you that this is variable and it depends on the position of the bubble. So there are some images that are easier to recognize, other images that are harder to recognize at the physiological level, and the same thing happens at the behavioral level. So at the behavioral level we can measure performance as a function of SOA and measure the area under the curve as an indication of how easy it is to recognize that particular image. So we did a whole psychophysics experiment with data from one of our lectures. So we took the exact position of the bubbles from that experiment, and we then conducted a psychophysics experiment inspired and guided by the position of the bubbles in the physiology experiment to measure the behavioral reaction times or the behavioral effects of masking, in this case, for each of the same images. And we found that there is a weak but significant correlation between how difficult a particular image is and the behavioral reaction times. Again, this is at the single image level, and mind you, we were looking at the two completely different sets of subjects. So despite all of this variation, there's still a correlation between the behavioral world and the physiological world in this case. So we next turn our attention to the computational models and how these effects of pattern completion be explained using our understanding of visual recognition models. And we used a battery of well-established computational models for object recognition. At the top here, I'm showing one particular implementation of the type of models that Tommy Poggio has been developing for a long time now. In this particular case, this is the implementation by Thomas Serre. And then on the bottom, I'm showing you one relatively recent implementation of a deep network that in 2012 was the winner of in the ImageNet competitions, where you bombard these models with millions of images from internet, and then you try to tweak these parameters to improve the top 5 performance in these type of models. I'm not going to have time to do justice to describe these models in much detail. For people who have never heard about this type of models, suffice to say that these are purely bottom-up models. They involve the concatenation of linear and non-linear steps. And in the case of the top model, there are 0 free parameters. We're not tweaking the parameters based on the images. In the case of the bottom model, there are lots of parameters that you're tweaking in order to try to improve performance on a set of objects, and then you cross-validate on a different set of objects in order to try to improve recognition of whole objects. So we asked how well these models perform in recognizing the same images that we used in the behavioral and physiological experiments, and that's shown in here. So again, on the y-axis, we have performance, either of human or of models. And on the x-axis, we have the percentage of visibility. The black line here on the top is human performance. That's what I've shown you before. Pixels perform terribly, so if you just take pixels and you try to do object recognition with pixels, that doesn't work at all. If you do use HMax or what's called Pool 5, one of the intermediate levels in these deep convolutional networks, they perform better and significantly above chance, but they still struggle significantly when you're trying to extrapolate from these very, very small amounts of visual information. Even the top performing model at least called AlexNet, still significantly underperformed humans. So based on the behavioral and physiological studies, we conjecture that the problem was that the representation of these models is not robust to these significant degrees of occlusion, and that there's a need for additional computation. So just to give a hint for what's going on, this is a particular low-dimensional rendering of the representation of these high-dimensional models for each of the objects. So each point here corresponds to one of the objects that we tested. There are chairs and fruits and faces and so on. And all of this is in a very high-dimensional space with about 5,000 different features, for example, in the context of FC7. Instead of visualizing this 5,000 dimensional space, we used a technique called stochastic neighborhood embedding here to render all of those in a 2D plot. So here the open circles correspond to the whole objects. So you see that it's relatively easy to separate these different categories of stimuli based on this representation. And indeed, these feed-forward models work very well in separating and recognizing this type of whole objects. But when we use the partial objects, we see that the representation is cluttered and also very different from the representation to the whole objects, showing or providing some hints as to why it's very hard from these purely feed-forward models to extrapolate from these partial objects onto the whole objects. The computational distance, so we measure the distance from each of these exemplars to the category mean as a way of saying how hard it is for this computational model to recognize these partially occluded objects, how distinct those representations are. And again, we find that this with this computational distance is weakly but significantly correlated with the neural latency. So we look at each single image, each single rendering of these occluded objects. We measure how long it takes to recognize, to elicit a selective signal at the physiological level. That's on the x-axis. And we measure the computational difficulty for that image on the y-axis. And we find that there's a weak but significant correlation between the computational and the physiological data. So I argue that at the behavioral level and at the physiological level that there's a need for additional computations. I also argue that purely bottom-up models do not seem to be very well, and they significantly underperform with respect to humans in recognizing these heavily occluded objects. So we conjecture that the additional computations that we need involve recurrent computation, that you need to be able to make, in order to make this type of inferences, you need to be able to either use top-down and or recurrent information. And one of the main models that have been proposed many years ago to be able to do error correction or pattern completion is the type of Hopfield network roughly illustrated here. So these are all-to-all networks. So there's massive recurrence. They're connected all-to-all. And it turns out that under certain conditions, you can show that this type of models can be very good at pattern completion and at sort of building specific attractors and shifting the dynamics of the system from partial information onto these attractors. And I'm not going to have time to do justice to this. Some of you are probably very familiar with this type of models. If you're not, you can just think of this as basically a landscape in energy world where you have attractors that are defining the objects that you want to recognize. And when you start in the middle of nowhere, when you start with an object that's heavily occluded, the dynamics of the system are such that basically you attract the object onto one of these well-defined attractor states. Importantly, in this particular rendering, so this is a schematic of what I just told you. Imagine that we have 3 neurons. In the brain, of course, there are many more. In the models, there are many more. So each of these axes here now indicates the representation of objects by these 3 neurons. So in this case, we are trying to recognize 4 different objects, 4 different prototypes. And our representation of that object ends up, because it's heavily occluded, not in one of those exact prototypes, but somewhere in between, because it's heavily occluded. So the dynamics of the system will be such that that object will be pulled towards one of those attractors. So that's the intuition of what we're trying to accomplish. And importantly, in this particular implementation of this system, we have 0 free parameters. So we're not trying to tune parameters to the system. We take one of these feed-forward architectures that were trained based on images on the net. This is AlexNet and or the HMAX implementation. And at the top level of that hierarchy, we implement the Hopfield network. This Hopfield network has attractor states that are purely defined by the whole objects, and they are well defined according to an equation whereby you define the weights that are symmetric, and they're exclusively based on the features of the whole object. So we're not trying to tune and fit specific parameters. This slide is horrible. I apologize for that. It's very hard to see. There are lots of lines, lots of different models that we tried. Again, on the x-axis, we have the performance. On the x-axis, we have the percentage of the object that's visible. On the y-axis, performance. You have human performance there on the top. FC7 is highlighted there. This is the performance of the purely bottom-up model. And I'm showing you FC7 plus this attractor network. So we only have recurrency in one level of the hierarchy, and this already significantly improves the performance of these bottom-up models. We're still not at human performance level with this attractor network, but again, even without using any type of parameter tuning, just adding this recurrency and these additional computations on these feed-forward models, we can improve performance. We can improve performance even much more if we allow ourselves to train with these occluded objects. In order to ensure that we're not overfitting, we train on some objects, we test on other objects, so we do cross-validation across objects. But now we have this FC7 representation, and we add these recurrent connections, which are not dictated by the attractor model, but they're actually trained based on the object. We're trying to minimize the distance between the final representation of the object after all of these dynamics and the representation of the whole object. So if we do that, then let me go back to this 2D representation based on SNE, the stochastic neighborhood embedding. This is what I showed you happens at the beginning, and then over time, the representation of the partial objects, the field circles, becomes closer and closer to the representation of the occluded objects. And then if we measure performance as a function of the amount of visibility, we have this RNN model, which is now trained on the occluded objects, and which achieves a human-level performance. So we can recapitulate human performance for heavily occluded objects by just adding one step of recurrency that's trained on these objects. In addition to showing performance, we also looked at the correlation at the image-by-image level. So what are the mistakes that humans make? What are the mistakes that the computational models make? So for that purpose, we measure the performance of the RNN model versus the performance of human subjects on the exact same images. We can see that at the beginning, there's no correlation. As a matter of fact, there is an inverse correlation. And if anyone is interested, we can discuss why that's the case. And as we go through the different steps of these recurrent computations, the correlation at the single image level increases. The correlation is clearly not perfect. You can point to lots of images where the humans get it right and the computational model does not, and vice versa. But there's a significant increase in the degree of correlation between the model and humans. As a matter of fact, if we just average and you measure the average correlation between humans as a function of time step in this recurrent computation, you can see the increase in correlation that I was referring to. And the dashed line there is the correlation between humans. So if you ask one person to recognize objects, the heavily occluded objects, and you compare the performance with another object, the correlation is not 1. Humans are capricious devices, and they also have their own preferences, and they also perform differently. So here's the correlation between between different humans. This is where we are right now with these recurrent networks. So we're in, as a function of how much recurrency there is, there's a significant improvement in the correlation with humans at the single image level, not only at the overall performance level, but there's still certainly a gap in order to be able to explain totally the human-human correlations in recognizing patterns from partial information. So to summarize, I talked at 3 different levels. I discussed at the computational, at the implementation, and at the algorithm level some of the basic ideas about how we can make inferences from very, very small amounts of information in the context of visual recognition. So at the behavioral level, I argued that recognition is quite robust, that you can take out about 90% of information about an object and still recognize it. And I also told you that if you do backward masking, if you present the image very fast and you immediately put a mask, you can significantly disrupt performance. And we took this as a suggestive indication that you need additional computations, perhaps recurrency and top-down, in order to be able to recognize these heavily occluded objects. I argue that if we put electrodes inside the human brain and we look at activity in the inferior temporal cortex, we get neural signals that are robust to heavy amounts of occlusion, so we still obtain selectivity, but the signals are significantly delayed, and the delay is in proportion to the amount of occlusion and in proportion to difficulty, and also correlates weakly but significantly with the behavioral data. I then argued at the computational level that purely bottom-up models seem to be insufficient to fully explain our ability to make this type of inferences from partial information, and that we need recurrent computations to solve the problem of pattern completion. And showed you a toy example where we implement recurrency at the top of these bottom-up models and rescue performance in recognizing whole objects. So I'm going to stop there and then mention one more time Han Lin Tang and Bill Lotter, the 2 amazing students who have actually done all this work.